Hey guys, how's everybody doing? We're on chapter 9. Chapter 9 this week. We're going on genetics and heredity, and there's quite a bit of vocabulary, so that's what I'm going to focus on. Go ahead and share my screen. Okay. All right. Chapter 9. Okay. Vocabulary. Start with heredity. Heredity is the transmission of traits from one generation to the next. So in other words, heredity is referring to inheritance, the inheritance of different physical traits, different traits that make up your body. And those things don't only include your physical appearance, but they include your organ function, if you're lactose intolerant or not, your blood type, all of that. Then we have genetics. And genetics is the scientific study of heredity. Again, you'll notice the use of the word scientific in this definition because there's other types of inheritance and heredity that you can study, or other ways you can study it. I mean, even genealogy is kind of one of those. Okay, so I'm going to give you a bunch of vocabulary, and then I'll kind of start putting it together for you so you can kind of see how all of those terms apply to each other. So for now, be patient. I'm just going to give you a bunch of vocabulary. All right, alleles. Yes, that's how you say that word. Looks like alleles or something like that, but it's alleles. Okay, alleles are alternative versions of a gene. So for instance, hair color, eye color. I'll just leave it at those two. Actually, I won't. I'll give you a couple more. Attached earlobes. So earlobes that kind of go straight in versus hanging down a little bit. Anyway, all of these are alleles. They're all different versions of a trait. So everybody has genes for hair color, but there are different versions of those genes, right? All the different colors of blonde, of black, of brown, of red. Those are the alleles, okay? Another term is the word homozygous. The same. Homozygous means that a person has two identical alleles for a trait. And so the homozygous genotype is represented by an uppercase, capital letter. Okay. Then we have the term heterozygous. Hang on a second, I just got ahead of myself a little bit. I'm trying to give you these other terms. Homozygous. You have homozygous dominant, meaning having two dominant alleles, right? And again, the dominant allele is what's represented by an uppercase letter. So first I'll look like that. We have homozygous recessive. Alleles for a trait. And that's represented by lowercase letters, so it look like that. Okay. Again, I'll put all of this together shortly. Just get through vocabulary. All right, then we have heterozygous. Hetero meaning other. Heterozygous is two different alleles. So somebody that is a homozygous dominant for black hair has black hair, and both alleles, the one from mom and the one from dad, both carry the information to make black hair. Okay. Someone that's homozygous recessive for red hair gets one allele from mom, one allele from dad, and both of those alleles have the instructions for making red hair. For somebody that's heterozygous, they have one dominant allele from one parent and a recessive allele from the other parent, okay? So their alleles have information for making two different hair colors. Which one comes out on top depends on which one has the dominant gene, okay? Because somebody that's heterozygous, we represent as looking like this. One dominant and one recessive allele, okay? Normally, darker hair colors are dominant to recessive hair colors, or to lighter hair colors, but hair color is actually determined by a lot of different genes, not just one. However, it's easy to think about because we can all picture different hair colors easily, okay? All right, moving along. So again, I'm going to talk to you a little bit about dominant alleles. Dominant alleles are always expressed. Always. You see it all the time. When it's in a heterozygous person, you see the dominant allele, not the recessive one. And that's because with recessive alleles, they're only expressed when there are two recessive alleles together. Okay? All right. Let's do phenotype. Phenotype is the physical appearance. PH for physical, PH for phenotype. Phenotype is the physical appearance. Then we have the genotype. Genotype is the genetic makeup. Okay. So your genotype determines phenotype. Your genetic makeup, your mix of alleles determines what you look like and how your body functions. Okay. All right, I'm actually going to pause there at all of this vocabulary because I'm about to take us into the world of Punnett squares. Remind everybody what a Punnett square is. A Punnett square is something that gets used to identify possible outcomes for allele combinations. Okay. We do Punnett squares. We have a monohybrid cross, which is comparing two individuals for one trait. One trait. So in other words, if you want to know, a possibility of you and your significant other having a baby with black hair, okay? So that's two individuals, you, your partner, and the trait is hair color, okay? Then there's something called a dihybrid cross, which I'm not going to demonstrate in this class. The dihybrid cross compares two individuals or two traits. So that would be like if you wanted to know possibilities for hair color and eye color. Okay. I'll give you, I guess, one more. A word. I like I just made that up. Okay. So Mendelian genetics, the alleles follow the basic rules of either being dominant or recessive. So in other words, it's either or. Got my big blanket I got on the chest. Okay. Because not everything does that. Not everything in our body follows the typical or the standard Mendelian genetic principle where dominant is always expressed and recessive isn't. Do I go over that in this chapter? I do actually. I'm just going to do it right now. Why not? Okay, so when this doesn't apply is in something called incomplete dominance. That's where you get the phenotype right? So incomplete dominance, this is where instead of having your dominant allele always expressed, you can get a blend. So with the example of snapdragons, type of flowers, you know, red is usually going to be dominant, okay? But when you have a red snapdragon that pairs with a white snapdragon, or their crossbred, the color is a blend. It's pink, so you don't get red and you don't get white. You get this intermediate phenotype. You get pink. Okay. Another example is something called codominance, and that's where both dominant alleles are expressed. An example of that is our blood type. So you know that humans, sorry, my phone, humans can have type A blood, type B blood, type AB blood, and type O. Okay? So A is dominant to O. B is dominant to O. AB is dominant to O. But if you have AB together, they are codominant. So that's why you have the blood type AB. Okay? So again, if you have A paired with O, the A is dominant. If you have B paired with O, the B is dominant, meaning you'd have B blood type. But when you have A and B together, your blood type is AB, codominance. Okay? So not everything in genetics follows that Mendelian inheritance pattern where it's only dominant versus recessive. Okay? Okay. Square time. All right. So let's go ahead and do some examples of Punnett squares. I don't know if you guys remember this from high school, if you did it in high school, but if you did, here's the review. If you haven't, I'll introduce it to you. So we do a Punnett square. There's our square, and we're going to divide this into 4 internal squares, okay? Let me move this out of the way. All right, so what we're going to do is we're going to do freckles. Let's do freckles, because that's a Mendelian trait, meaning it follows the either dominant or recessive. So freckles being a dominant trait, I'm going to give a capital F, because remember we do capital or uppercase letters for dominant traits. And then for somebody without freckles, we're going to do the lowercase f, okay? So let's cross a dad who has a genotype of this. Ask yourself real quick, or see if you can answer what this genotype is before I tell you what it is. This is homozygous, right? Because there's 2 of the same. And they're both dominant alleles, so this is homozygous dominant. okay? You will need to be comfortable with these terms because on the quiz and then on your final, I'll give you questions that say like, you know, if you cross one person that's homozygous recessive for a trait with another person that's heterozygous, what are the possible outcomes? You need to remember what homozygous recessive and what heterozygous are so you can make yourself a Punnett square. For now, we'll go over like this, okay? So we're just gonna put one parent at the top and we put one allele over each square on the top, okay? And let's cross that with mom, who's also homozygous, right? Two of the same, but she's homozygous recessive. Okay. That means now we're going to put one allele down the side. So we've got one parent across the top, one parent down the side. Now we're literally going to cross them. This F with this F, right? Now this F with this F. Top with this bottom side, and last one on the right, bottom left, okay? Now, what do we end up with? What are all of these offspring? Or what this is called? We have one of each. It's heterozygous, okay? So the offspring for this type of a mix will have a 4 out of 4 chance of becoming or being heterozygous for this trait, okay? When you look to see what the possibilities are, you measure them in ratios. Let me just say. Okay, so remember our words, phenotype and genotype. Phenotype, again, is the physical appearance. So now we have a phenotypic ratio, and then we have a genotypic ratio. Okay, in other words, we're comparing the different phenotypes to each other, and then we're comparing the different genotypes to each other. Okay, let's go back and remember that our phenotypes that we're talking about here were either freckles or no freckles, right? So in our phenotypic ratio, we want to look at how many of these individuals, how many of these offspring potentially, we're going to have freckles versus how many are not. Okay, if we look at this, we can see that, well, all of them are going to have freckles. Why is that? See if you can answer it for a second before I tell you. Why do all these guys have freckles? Remember that genotype determines phenotype. With that being the case, the fact that they all have a dominant allele means that they're all going to have freckles. Yes, they have the recessive allele, but they're not going to see it, right? You're going to have freckles anyway, even though you have that recessive allele. So our phenotypic ratio in this case is basically 4 to 0, okay? Meaning all have freckles, okay? But what about our genotypic ratio? There are 3 genotypes, which I've put in the notes, but I'll make it more distinguishable here. 3 genotypes: homozygous dominant, homozygous recessive, probably guessed the third one at this point, and heterozygous. Okay, well, how many homozygous dominant individuals came out of this mix? None, 0. How many heterozygous individuals are there? 4, nice. How many heterozygous recessive individuals? None, okay. So our phenotypic ratio is 4 to 0 because they all have freckles, and our genotypic ratio is 0 to 4 to 0, okay, because we have only heterozygous individuals. Let's do another one. And I am going to use the same thing. We're going to still use freckles. But this time, our parents are going to be heterozygous, both of them. Remember that F is freckles. Lowercase f is no freckles. Okay? Parents are heterozygous, meaning that their genotype is that, right? One dominant and one recessive allele. I'm going to put one parent across the top. I'm going to put one parent across the side, and then I'm going to match these. I'm going to cross them. Okay, so here I get two big Fs. Here I get a little f and a big F. We're still going to write the dominant allele first on all of them. Here, another big F, little f, and here I've got two little f's. Okay, I'm going to write out here, do genotypic ratio this time first. And then we've got our phenotypic ratio. Again, looking at how many of each and a ratio comparing how many of each we have. Genotypic ratio. So again, we're looking for homozygous dominant, heterozygous, and homozygous recessive. Okay, well, how many homozygous dominant individuals do we have? One. Okay, so we have one person that's homozygous dominant. How about heterozygous? Two. There's two of them. And now, what about homozygous recessive? One. In a heterozygous cross, when you have two parents that are heterozygous for a trait, the genotypic ratio is always 1 to 1. Okay. And if you remember that, then it'll save you a ton of time when you're doing exam or quiz questions, or yeah, you won't have to like draw out a Punnett square. You'll just know if you have a question about genotypic ratio. Similarly, for a heterozygous cross, the phenotypic ratio is also always going to be the same. So now what we want to do is, let's see if we can figure out who has freckles and who doesn't. So how many individuals on this Punnett square outcome are going to have freckles? 3. These guys, right? This guy's homozygous dominant. These two are heterozygous. They have the recessive or the dominant allele. So all 3 of them are going to have freckles. Okay. But how many are not going to have freckles? 1. Okay. So your phenotypic ratio is always 3 to 1 for a heterozygous cross. Okay. Okay. When we're doing a Punnett square, though, I'm just going to throw this back up. That was terrible. Now it looks like, you know what? You know what? I'm going to change up my letters here because I'm just doing this as an example. My Punnett parallelogram. Okay, we're going to go with A. It's easier to tell. Okay, so I'm going to give ourselves heterozygous parents again. Okay. The point of the Punnett square is not to tell you what you will get. Okay, it tells you what you could get. It's a probability thing. Okay, so if I were looking at offspring, and let's take it away from people for a minute, and let's say that I'm looking at coat color for dogs. Okay, and I want an idea of if I breed this male with this female, what are my possibilities? What is the probability of me getting a certain coat color? Okay, that's where the Punnett square comes in. And if they're both parents are heterozygous, then we know that we have a possibility of having 1 out of 4 being homozygous dominant. You know, you've got a possibility of 2 out of 4 being heterozygous, and you know you have a possibility of another 1 out of 4 being homozygous recessive. Okay, what does this matter? Well, if we take this away from animals and hair color, and I make this something that's more life-threatening, something like cystic fibrosis, which is abbreviated CF. Now it has a whole lot of meaning. Okay, so cystic fibrosis causes a mucus buildup in the lungs and in the digestive tract. It's painful. It causes severe problems with breathing, prone to every type of constant frequent bacterial infections because all of that mucus makes it a happy place to live. Most people with CF have shortened lifespans and restrictions to their ability to live. It's a big deal. It's one of the ones we try to figure out how to eradicate genetically. So cystic fibrosis is something called a recessive disorder. And yes, these are part of your notes. I'm just writing them. Recessive disorders mean that they're carried on the recessive allele. Well, the thing about recessive alleles, you have to have two of them for them to be expressed, right? So let's say that I'm a parent. I am a parent, but you know what I mean, hang on. No, CF, okay. And let's say that, let's say I know I'm a carrier. What's a carrier? Let me give you that vocabulary real quick. A carrier is someone who has the allele for a trait, but does not show it. Okay? So in other words, I can be a carrier for a recessive disorder, but not have it. I have the allele, but I don't have the disorder because my dominant allele protects me from it. Okay? So let's say that I know that I'm a carrier. And now let's say that I know that my husband's a carrier too. That gives us this genotype each. We both would be heterozygous, meaning that we're both carriers for cystic fibrosis. So if I do this cross, this is going to help me know what my chances are of my husband and I having a child that has cystic fibrosis. And I can see that when I look at this, I have a 1 out of 4 chance. This isn't actually me. I'm not talking from my own experience. I don't have the trait, the allele for this. I'm just using us as an example. This would mean that we'd have a 1 in 4 chance of having a child with cystic fibrosis. I'd have 50% chance of having 2 of my kids be carriers. Okay. What happens if this was us and we have kids and we don't do the genetic screening in vitro, we just have kids. And then we end up having our first baby that has cystic fibrosis, okay? That doesn't mean that I'm never gonna be at risk of having another child with CF. Because the thing about Punnett squares and genetics is that every single time is like rolling the dice, right? Just because you rolled a 6 this time doesn't mean you're not gonna roll a 6 the next time you roll it, right? It starts over. Well, that's what happens with genetics. Just because I had a child first that had cystic fibrosis wouldn't mean that I now only had the options of having 1 that's homozygous dominant or 2 that are heterozygous. I still have a 1 in 4 chance with every child that I have. And again, this isn't my experience. I'm using me as an example to make the point. Okay, so it starts over every single time. And that's why you have to remember this as being a probability thing, again, similar to rolling the dice, okay? Now, let's look at what happens if I'm dealing with a dominant disorder. So a dominant disorder is going to be found on the dominant allele. Okay, So there's the disorder called PKU, and it has a problem with a certain type of amino acid. PKU is fatal in infants if it's not caught immediately, which is why they do a blood test at birth for it, because it's easily not treated, but it's easily handled. You put the baby on a special diet, special formula that doesn't contain that particular amino acid, and then they're fine. However, if it's not caught, if it's not treated, the phenylalanine builds up in the brain and causes death within days to weeks. So it's a dominant disorder. So now I'm going to say that capital A, carries the allele for PKU, whereas lowercase a is, let's say normal, we'll say no PKU, okay? All of a sudden, our chances are different, right? Because before, when it was a recessive disorder, we only had a 1 in 4 chance. But now that it's a dominant disorder, look what happens. Now I have a 3 out of 4 chance, right? This person now is the only one that is not going to have it and has no ability to have it. These 3 genotypes all would have it. Because again, even though the heterozygotes have the recessive or normal allele, it's not going to be expressed because of the dominant allele, okay? You know what? I just used the example of PKU, but that's a recessive disorder. I don't know what I was thinking. Forgive me, I'm going to switch this to a different type of disorder. Let's do achondroplasia. Achondroplasia is dwarfism. It's like those odds are too high for PKU. That's not how that works. I don't know why I'm writing it like this. Probably because this is my 5th lecture today and I can't think of impressive words. Okay, this is better. All right, so again, in dwarfism or achondroplasia, a 1 in 4 chance of having a child that is not affected by it, does not have it, but a 3 out of 4 chance of having a child that is. That's why when they talk about people with dwarfism being able to have offspring that are normal height, this is why. It's because of this 1 in 4 chance of having somebody that's recessive for it, okay? But a 3 in 4 chance of having somebody that has it. With dominant disorders, there are no carriers because you either have it or you don't, okay? So in recessive disorders, the heterozygotes are carriers, right? They don't have the disease, but they carry the allele for it, meaning they can pass it on. But in a dominant disorder, you don't carry it. It's either there or it's not, okay? Talked about disorders, but this is also true for traits. Again, I'm oversimplifying it because hair color is determined by many genes, not just one, so it's not purely Mendelian. But again, oversimplifying, using myself as an actual example this time, me. Don't know if you've noticed, I have red hair. Both of my parents had red hair. My sister, my only sibling, has red hair. We have red hair. That means that my genotype, if I'm going to oversimplify it, would look like this because it is a recessive trait, okay? So I look like that. My husband is Mexican, if you haven't guessed by my last name, Gonzalez. I know I look like one, right? I look like Gonzalez. I don't. My husband has black hair. Normally that's a dominant trait, okay? So I'm going to assume that he'd be this, okay? So what would happen is if I did just basic Mendelian genetics and did a Punnett square, all of our kids would come out being heterozygous, right? Because I only have a little A to pass on and he only has a big A to pass on. Only have recessive, he only would have dominant to pass on. That means our kids would look like this. right? If this was actual Mendelian genetics, then I would expect all of my children to have black hair. Because there are multiple alleles and multiple genes responsible for actual hair color, my kids all have brown hair, varying degrees, varying colors of brown. My son, my oldest, I love it. My genes are fighting hard in there to be expressed. His hair is brown, but right here in the front, where it's long, it's red. Like, no lie, maybe it's mixed in with the brown, but it's impressive. I'm like, way to go, red hair genes. You fight, you fight. My point, my children are carriers for this gene. Okay, they're carriers for this trait, I should say. They're carriers for red hair. Meaning, I could end up with grandbabies that have red hair. That's what I'm shooting for. I will have to arrange their marriages based on their chosen person's genetic profile. I will need to know what their hair color is and what their genotype for hair color, all the different genes are, and then I can determine the likelihood of having red-haired grandchildren. That basically means that I have to go with somebody that has lighter hair, like the blonde to light brown, if I really want to increase my odds. So I've already discussed it with my kids. They're good with arranged marriages. They understand that I need to pass on the red-haired gene. Got to keep our people alive. And so they're fine with it. Not really, although it is a joke. I know as my kids get even older, they're going to be like, Mom, she's got dark hair. Are you going to let her come in the house? And be like, no, she can't come in the house because she doesn't have red hair. You got to have red-haired grandbabies. Or he. I do have a daughter. I do have a daughter too. So anyway, how genetics works. This Punnett square is also really useful in being able to tell why you and your siblings have similar traits, but you're not identical. Because you can see that if you did a Punnett square for every single trait in your body, I mean, you'd have a gajillion Punnett squares, but you'd see the unique combination that results out of every single one. And so you and your siblings have the same mix of genes that are going into it, but what comes out every time is different. And again, that's also part of the crossing over and independent assortment and random fertilization that we've talked about in the past. Okay? That's it. I condensed this lecture, made it a little bit shorter than usual, believe it or not, and you're done. You are done. That's your lecture this week. The quiz will be up. Yeah. Talk to you soon.